Hi everybody, it's me, Mr. Olson, and we are going to be doing our final review session. This one will be done only online, and it's just going to go over genetics, things that we've already covered in class pretty well. And we even did it earlier this week, so it should be fresh in your head, but just in case you feel like you want another look at it, well, here we go. Let's do this thing. All right, so let's get some basic terminology out of the way. We have this term dominant. Dominant is going to represent any allele that shows, even if there's another recessive allele available. If you guys remember what we were doing in class, we basically said a letter that is, just a capital representation of that letter, whether it be an A or a B or a capital C or a D, it doesn't matter. Those represent dominant alleles, so they seem to take over whenever they're paired up with another letter that's recessive. Now, a recessive allele, these things are going to be alleles that are shown only if the two copies appear. A good example of that would be a little a and a little a. That is the only way you will ever see something look recessive. So that's actually a recessive phenotype. Then we have the terms homozygous and heterozygous. Hetero means different. So we're saying these alleles are different from each other. So you have a big and a little. And try to remember, you can notice here, you can see the big letter always comes first. Never, ever, ever put the little letter up front. And then we have a homozygous organism. And they can be homozygous dominant or recessive. And these are when the alleles are the same. So that's either big A, or little a, little a. Doesn't really matter the letter or what allele it is. Then we have genotype and phenotype. You should remember that genotype is the two allele letters showing what genes they are. Any one of these things right here, any one of these guys, those are examples of genotypes. But a phenotype is the physical description of what those alleles actually express. Does it look tall? Does it look short? Is it a purple flower? Is it a white flower? Anything that it actually looks like, that's what we'd use the word phenotype to describe. Now, there were a few laws that we talked about. The law of dominance is the rule that states that the dominant allele takes over when it's paired up with the recessive allele. So if you remember us just saying just a few seconds ago, big A, little a, this guy's going to win. That's because it's the dominant allele. It seems to take over. It makes it seem like the recessive traits seem to disappear from subsequent generations, but the truth is they definitely do show up later on. As long as they have another recessive allele show up. So let's go ahead and talk about some different ways that you can do genetics. The first method that we used in class to learn how to do genetics was really called Mendelian genetics, but I guess it's more technically referred to as complete dominance. And I want to just work through one or two problems with that of that style for you. So it says, what phenotype do heterozygote genotypes have? This means if you have an organism that is big A, little a, what is it going to look like? So overall, the rules should look like this. We have a big A, little a, and, sorry, a little a, little a. So these are the genotypes, okay? Let's go ahead and pretend right now for this example, big A is going to represent, I don't know, let's say red apples. I'm totally making this up. And then a little a is going to be a green apple. Okay, so the big A is dominant to the little a, so red apples are dominant to green apples. So the genotype is the letter combo. The phenotype, in this case, big A is going to look red. And the bottom case, it's going to look green. But in the middle case, of the two letters here, which one wins? That would have to be the big A, which means this would look red. So you should be able to tell what phenotype we're going to have based on the genotypes with the rules of complete dominance. And once again, on the exam, if I ever ask you a question, I will tell you which rule set to use. I will say, please use the rules of complete dominance or incomplete dominance or codominance. So just to give you an example, if I cross a big A with a big A, and a little a with a little a, so I have a red parent that's homozygous dominant and a recessive parent that's homozygous recessive. In every single one of my boxes here, I'm going to have big, little, big, little. If I look at another example, let's say two heterozygotes are going to be crossed. Both of these parents look red, but they have a kind of a hidden trait for the green feature. So it looked like this. And we use some interesting phrases like a genotypic ratio. And in this case, a genotypic ratio, it would be a 1 to 2 to 1, because we have a big A, big A, a big A, little a, and a little a, just of the number of boxes that we have. And then we have a phenotypic ratio. And this is what the physical appearance looks like. So we're looking at a ratio of that. Well, 3 of them are going to look red, and only 1 of them is going to look green. So that would be red to green. And that's kind of what you do for genotypic ratios. I might say, what's the percentage of the children that are born red? Well, if that's the question, then you look at your square and you say, okay, this is red, this is red, this is red, this is not red. So that's 3 out of 4, which is 75%. And if you ever work with a monohybrid cross, just one of these small, normal-looking Punnett squares, your probabilities of finding something are either 0%, 25%, 50%, 75%, or 100%. So it's a little bit easier to work, and we work through a good number of these problems in class, so I don't feel like you'll mess this up at all. I feel like you all feel pretty confident about it. But let's go ahead and try another rule set. This brings up a dihybrid cross. You can see the letters for dihybrid crosses means 2 allele sets will happen at the same time. You could have parents look totally different, like the different alleles don't actually affect each other, but it could result in a different phenotype, basically showing that the 2 traits happen at the same time. So let's say we wanted to set this up. You could use the FOIL method, F-O-I-L. So let's say I take my first parent right here. F would be first letter, which would be big A, big B. O would be outside letter, so outside letter, outside letter. So that would be big A, little b. Inside would be little a, big B, so little a, big B. And last would be last A, last B, so little a, little b. But I came up with my own thing. You could also do FOIL on the side here for the other parent. But if you're not going to remember those things, because maybe you just have a bad taste in your mouth from math and you don't want to use that method, okay, that's fine. You can also use the code 13, 14, 23, 24, and you just number each letter, and then you just attach which letters go there. So you can just kind of work it that way. It's your call. It seems a lot messier that way, but it also is more deliberate that way, and you'll be less likely to mess up. But the truth is, we don't even like this method. Like, this method's too big, it's too slow, oh, it's a pain in the butt. And then once you get all your letters written in here, and you'll have like 4 letters all squeezed into a box, and then you gotta go through and see which ones you're looking for that actually have the trait combination that you're looking for. It's not very good, but I like this method a lot better. All you got to do is just 2 simple Punnett squares. So let's say I have a parent that's big A, big B, little b. I want to find out what happens if I cross it with a, big B, big B. So let's say there's a question on the test, and it says, what percentage of the kids will be born big A, let's say big B, big B? Well, it's easy to figure out. So first thing I do, I'm going to do these in different colors for you. I look at my A's. So over here, I do big A, and the other parent, little a, and I cross it up. Okay, so I'm looking for this. So what's the percentage of kids that are born this way? That's a 2 out of 4, which is a 50%, by the way. And then for the other parent, I'll do it in green. I got my B's and my B's. So I got a big B, little b, big B, big B. Cross them up. And then what are my odds of getting big B, big B? Well, if I look here, these 2 boxes, which is a 2 out of 4, all I got to do to figure this out, to find out this answer, is to simply take 2 over 4 and multiply it by 2 over 4. So this one, and multiply it by this one. And to do this, you just multiply across. There's nothing really fancy there. So 2 times 2 is 4, and 4 times 4 is 16. So 4 over 16 simplifies to be 1 over 4, which equals a 25% chance that we have a kid born this way. So you don't have to do a gigantic Punnett square. You don't have to take all that time. You just got to do 2 simple small Punnett squares, and life is easy. You could even do this for something that's like a trihybrid cross, or I don't know, a pentahybrid cross. Like, you can just have as many traits as you want, and you just do 1 Punnett square per allele set. So let's go ahead and talk about the tweaks to those rules. If you remember earlier, we talked about genotypes being big A, big A, big A, little a, and little a, little a, and we said they look a particular way. So let's say in this example, I think we're going to go with, I don't know, let's go with flower color. So big A, we have a red flower. Little a, we have a white flower. And in this case, if we ever see big A, little a, we're going to say it's a merging of the 3 colors. Basically, we have a new third phenotype that can show up. So this would be red, and this would be white. And if we ever see a heterozygous organism show up, it's going to be a blend of the 2 different phenotypes. So let's go ahead and say, what happens if I cross a red parent with a white parent? Hey, I've suddenly changed letters. I apologize. But we're going to rock that anyway. Should have been doing this anyway from the beginning. Well, every single one of these is going to end up as a heterozygote, and the percentage of this being pink, well, that should be pretty easy. It should be 100%. Every single one of these will be a pink flower. If you were to take all heterozygotes, you'd end up with another, oh, sorry, my bad. You'd end up with another interesting ratio. If we were going to look at a genotypic ratio, the genotypic ratio would be 1 to 2 to 1. If we look at the phenotypic ratio, it's actually going to be the exact same thing. It'll be a 1 to 2 to 1. The reason for that is because now that we have this new third phenotype that matches up with this genotype, we end up actually seeing genotypes and phenotypes match up. The next one we talked about was codominance, and yeah, it says what phenotype do heterozygous genotypes have. So let's say if we were going with red being red, and now another letter, we use the W to represent white, because we're saying both of these are capital letters, so they're both dominant. That's what codominant means. If you were to have a heterozygote, which means 2 of the same, 2 of different letters, this would mean the flower, in this case, would look both red and white at the same time. It would not look pink. It wouldn't be a blend. They'd be 2 distinctive colors at the same time. So you'd see spots of white, spots of red. Yeah, I would say it's a very spotty looking physical appearance for the phenotype. Anyway, if we were to cross these up, let's say we crossed a red flower with a red and white flower, well, you'd see a majority of these flowers, actually, we couldn't say that, could we? Half of these flowers would be red, and half of them would be a mix of red and white. If we were to cross 2 heterozygotes, you'll see the same relationship that we talked about earlier. Much like what we saw with incomplete dominance, you will definitely be able to tell we have a new third phenotype, so a genotypic percentage. We're going to see a 1 to 2 to 1 ratio. With a phenotypic ratio, we will still see a 1 to 2 to 1 ratio. And if it asks you a question, you know, what percentage of these children in this one here, which one are going to be red? Well, it'd be easy. It'd be 1 out of 4 boxes, which is 25%. So on your midterm, yeah, you're going to have genetics problems. And yeah, you should use some scrap paper to work through them every time you see a problem. Don't just do it in your head, just to make sure you don't make a mistake, even if it seems easy. But you should also make sure that you actually have read the question and you know what they're asking you for. Are they asking for genotypic ratio, or are they asking for a percentage? So try to keep your eyes peeled for that. There are also things out there called sex-linked traits. This would be a feature that is unique to which sex chromosome an organism has. So we talked about females having XX, and we talked about males having XY. But the funny thing is, the X chromosome is really big, looks kind of like that, or is the Y chromosomes really tiny? So this thing can't hold very much stuff at all. It's too tiny. So traits seem to stick to the X chromosome. So it kind of asks this question, why can only females be considered carriers? It's because females, they can come in 3 versions. Let's go ahead and say we're going to use big R. No, a good example of this is actually color blindness. So let's say big B is normal vision, and little b is the person or the organism will be colorblind. Not blind, but colorblind. So they can come in 3 versions. They can look like this, this, or this, which means this is a female that's, you know, blind, colorblind. This and this, well, the top one here, of course, they have normal vision. This woman here would have normal vision because the big letter wins, the big B wins, but they're considered a carrier because they carry the trait, but they don't express it themselves. But their kid could end up with it. With the male different genotypes that we have XY and XY. There's only 2 possibilities. Either the B carries something and it's a dominant, or it carries something that's a recessive. The Y chromosome is so small that it literally can't carry these traits. So it has to, the male has to express whatever's found on the X chromosome. So in this case, the male will have normal vision, and in this case, the male will be colorblind. So let's say we cross a, let's say it's a colorblind female, which is pretty rare, and we cross it with a normal vision male. If we end up doing this, there's a 50-50 chance that the child would be born a female, and this is actually supported by this graph because there's only 2 boxes that'll have an XX. My handwriting's terrible. So these females would all be normal vision, but they would all be carriers as well, which means their future children would have a chance of being colorblind. And for the males, it's a guarantee that all the males that would be born colorblind. Remember, the Y chromosome is too small. It can't hold anything there. So that's basically all we have for that one. It's just always attached to the sex chromosome in this case, or an X-linked chromosome. There are actually traits that only stick to the Y chromosome, but they're exceedingly rare, and we didn't really talk about them in class, so you don't have to worry about those. Lucky for you, we are not going to have anything on the test about pedigrees because we simply just didn't have enough time to get there. But let me go ahead and take the time to mention another form of codominance, and that was called blood typing. If you remember, you know, if you've ever had your blood drawn, you can have A positive blood, AB blood, B negative blood, O positive blood. Like, there's positive, negative versions of everything, but there are really 4 main types, and that's what we're talking about. We have A, B, AB, and O type bloods. The reasoning behind that is because of the proteins that are kind of associated with each type of blood. They actually kind of hate one another, so they don't mix together very well, which is really important for, you know, medical professionals to know. But for our purposes, we're just going to look at it in terms of genetics. So we usually refer to this in this way. We use these capital letter I's to represent this, but A-type blood can come in 2 forms. You can have a capital I with an A, and a lowercase I, or you can have 2 capital I's with A's. This one would be homozygous for A-type blood. This one would be heterozygous. The I represents O-type blood, which means it doesn't have a dominant feature at all. B-type blood is set up the same way, but it uses, instead of a dominant B, instead of a dominant A, it uses the dominant B. So it could be set up as a homozygous or a heterozygote. AB-type blood is very interesting because it has both an A and a B, which actually means they don't fight each other because they don't have the components that make them hate each other. So they actually get along with everything. Fun stuff, we actually said that O-type blood is considered the universal donor. If you are O negative, as a matter of fact, your blood can be given to anybody, vice, you know, some very interesting things that might happen to your blood. And then AB type blood is the universal receiver. And their whole thing is that they could take blood from anybody, whether it be O type, A type, B type, AB type, because they don't have anything as a component of their blood that fights that. So there's only 6 different genotypes here, but it looks really confusing. Sometimes people will be lazy and kind of write it this way. I don't know if this helps you or hurts you. If you don't understand, or if you can't look at it and understand, then don't worry about it. Like, you already know the method that we used in class. But you could say you're still homozygous for A-type blood, or heterozygous, homozygous, heterozygous. You're still heterozygous for AB, and then you're, of course, homozygous for O-type blood. The O's would just be a recessive trait there. Of course, when you write it out like this, it doesn't help you recognize that the little I is a recessive trait. That O type is a recessive trait. This would just make you think there's 3 dominant alleles. But anyway, let's work through a problem here. Let's say I have a parent who's homozygous for A type blood and heterozygous for B type blood. And what percentage of kids are going to be, let's say, A? So I don't remember what I said, but I'm pretty sure I said a parent that's homozygous with A type blood and heterozygous with B type blood. So then you just cross this up. It's always smarter to do your big letters first, then you can just fill that in later. It's a little I. So when you look here, what percentage are going to be A-type blood? Well, this one, and this one are both A-type bloods. So we'd say there's a 50% chance that they'd be A-type blood. There's no chance that the kid would be B-type blood. Yeah, I know some of you might look at this and think, oh, well, there's a B right there, Mr. Olson. Like, that's B-type blood. No, remember, if you see both an A and B at the same time, that makes AB-type blood. So don't get that confused. If it asks you for a particular type of blood, there are 4 different types: A, B, AB, and O. So try to remember those differences there. We had some fun questions that we worked through using these very things. So that's pretty much it. If you guys have any final questions, feel free to ask me. If you guys remember your schedules for this exam week, we are getting a study period before every exam. So that means when you show up to class, we'll have about 50 minutes, 50, where you are studying in class. And if you have particular questions on anything, I'm going to be walking around. I'll be helping people and just seeing if you need help with anything. This would be a good time to work out some things, maybe practice some stuff, maybe quiz with a neighbor, and just try to get comfortable with the material as best as you can. Bring study guides or resources that you've been using, and yeah, help each other out the best way you possibly can. And I'll be there helping you do the same thing. Before we call it quits, I just want to go ahead and take a moment to give you some test-taking tips. I know midterms and finals, it gets kids pretty stressed out. Kids don't like taking big tests. Nobody likes taking big tests. But let me just give you some advice, okay? First off, don't just cram the night before the test. If you're watching this video and it's 3 a.m. Monday morning and your test is in 4 hours, you're doing it wrong. You should have been studying well before this. There's actually scientific studies that actually support our claims that say, you don't need to go out there and cram, because it doesn't actually help you. It doesn't actually support anything you're doing there. Don't lose sleep. Actually try to get some rest that night. Even if you're nervous, just sit down with a tea or something beforehand, a nice hot cup of milk. I don't know, just do something, read a book. Just try to get some rest, guys. Don't stress yourself out. Eat breakfast if you can when you wake up. Just a bowl of cereal, a granola bar, a piece of cake. I don't really care what it is, as long as you get some sugar in you to get your metabolism going, get that brain moving, and hopefully you'll be thinking a lot better when you sit down and take this test. Do the best you can. I know I can stress out pretty easily for myself, but a good piece of advice would be simply to just relax as best you can. This test is not the end of the world. This doesn't mean you're going to fail the class. It doesn't mean that you're you're a bad person if you don't do well. Some people just notoriously don't do well on tests because they stress themselves out. But remember, when you're taking the test, relax. You have plenty of time to take the test. You're very capable of doing the test. I just want you to try your best so I can see, kind of what you know and what you may not know, and that will help us in the future when we prepare for the EOC. So just take your time. I'm not trying to judge you. I'm just trying to figure out what you can do on a piece of paper. It's not a judgment of who you are. And remember, you have over a minute per question. Most kids will probably take 15 to 20 seconds per question. That means you could read this question over multiple times, read over your answer choice multiple times before you even decide to bubble in an answer. And these are all multiple choice. Like, you don't have to worry about long essay responses or short answer. There's no matching, so there's no metagame involved to figure out where things go. You are simply just trying to put one of 4 letters in a bubble, and that's it. There's even another reason to not stress out, because you get an extra credit question. That means there's no penalty if you don't do it. There's no penalty if you get it wrong. And as a matter of fact, you can get partial credit. So if you have this extra credit question, it is going to be an essay form, and that means you can earn up to 10 extra credit points based on your answer for this. If you don't realize it, 10 extra credit points is literally a letter change in your grade. So if you have like a 79 and you answer the essay question perfectly, you could end up with an 89. Now you have a very high B. Even if you just get a few points and you had a 79, you could push it over and get that very coveted little B that you wanted. So, just try to do your best, try to relax, and just use some good test-taking strategies. I'd also recommend, since it's a long test, why don't you slow down when you're bubbling things in? Be aware if you're skipping a question, mark things for review so you can come back to them, and just make sure that you don't misalign your bubble choices with the question you are on the test. I don't know how many times I've seen it, but you got a kid who's bubbling in number 24 but forgot they skipped number 23 because they didn't know the answer at that time. And then that causes all the questions that they bubbled in at the end to shift one place, which is a pretty significant error. And of course, when I grade it, I will see that you've marked every question wrong, probably, from that point forward. So make sure you go back, you take your time. I would say every page, stop and check to see if you have made a mistake and you need to fix your answer sheet. All right, so that's it. That's the end of review. We've done it. Thanks for listening. Thanks for watching. Thanks for using the guides online. You guys are a lot of fun to work with. And hopefully you've learned a few things for this first half of the year. Hopefully we've kind of encompassed those in the video here. And if you have any questions, once again, talk to me before your exam, and hopefully you do well. All right, guys.